Okay, so we will move to Lena, who just recently completed her PhD at ETH Zurich. She is in Cyreddy's lab and did her PhD in computational and systems immunology, where she developed statistical and computational modeling approaches for antibody specificity and antibody affinity engineering. Very much looking forward to your talk.
Thanks, Victor. Give me a second. I will start sharing. So does this work now?
Yes. Yes.
Okay, perfect. Thanks so much for the introduction, Victor. I'm actually really excited to be able to speak here today. I was a PhD in the Ready Lab, or the Laboratory for Systems and Synthetic Immunology at ETH Zurich, and I'll be talking about a part of my PhD research, which was on antibody affinity engineering using antibody repertoire data and machine learning. So Eric has already kind of like taken away a lot of the introduction on B cells, so I'll be like trying to keep this like very short. But we all know that antibodies are very important or a very important part of our adaptive immunity, as they bind to pathogens, and B cells are responsible for producing antibodies, and they essentially undergo affinity maturation, which happens when a B cell becomes activated and recognizes an antigen, and they will introduce mutations that will either or will eventually make those B cells present or produce high affinity antibodies. So if a B cell then essentially produces a high affinity antibody, they will further differentiate into antibody secreting plasma cells, which are essentially antibody factories that produce very large quantities of antibody that help us clear an infection, or memory B cells, which prevent future infections from the same pathogen. So we all kind of know how an antibody looks like. So I will just like briefly mention that, they consist of two heavy and light chains. And there's 2 or 3 parts that are particularly important on the antigen, which is the Fc region that mediates downstream effector functions and activates the immune system, which then essentially help us clear the pathogen from our system. And there is the variable region, which is responsible for binding an antigen. So we all have a huge diversity of antibodies in our immune system, which we call, like the set of those antibodies, we call antibody repertoires. And the genetic diversity in B cells encoding these antibodies is generated by VDJ recombination, which is a process that like many of us probably know very extensively. And particularly CDR regions are important as they form flexible loops that directly interact with the antigen. So this theoretical diversity of an antibody repertoire that can be created with these diversification mechanisms is estimated up to 10 to the power of 13, which I kind of want to emphasize on, because it's like really, really huge. If you would imagine that the Milky Way has 10 to the power of 11 stars. So when an antibody then binds a target or a pathogen, it can either directly or indirectly help to eliminate these pathogens. For example, an antibody can neutralize a toxin or a virus by blocking it from entering or interacting with host cells. Antibodies also activate downstream immune cells and promote phagocytosis or cell killing. And antibodies also activate the complement system, which is part of our innate immunity, that further downstream helps clear the pathogens from our systems. So some of these functions, or even just high specificity with which antibodies can bind to theoretically any particular target has made those very promising molecules for therapeutic applications. There's already a pretty large number of therapeutic antibodies out there on the market, and they're being used for a pretty large variety of diseases, such as, for example, cancer immunotherapy, autoimmune diseases, or infectious diseases. So while there's already many antibody therapeutics on the market to treat diseases, the development of a therapeutic antibody, however, is still pretty far from being streamlined. Overall, or a classic antibody development campaign roughly focuses on 3 stages. Antibody discovery is targeted at finding initial binding candidates to, for example, a known disease target. And this can be done either in vivo, where you screen B cells from immunized animals or humans, or in vitro, where we use display methods to screen synthetic libraries of antibodies. Antibody engineering involves optimizing an antibody's functionality, which could be, for example, to optimize antibody affinity towards the target antigen, or to reduce cross-reactivity to targets the antibody should not bind to. And lastly, one needs to optimize developability properties, which are antibody or protein properties that are unrelated to its functionality, however, yet very critical for drug development. And this could be, for example, stability or expression. So despite this extensive optimization, which requires a lot of experimental screening in this entire development pipeline, still less than 10% of the drug molecules coming out of this development pipeline will eventually be approved for the treatment of diseases. So the cost of this entire pipeline, including the high failure rate in clinical studies, substantially contributes to the high cost of treatment, which is on average $100,000 per year and per patient. And this is substantially more than with any other drug modalities. So you could imagine there is quite substantial interest in trying to improve the success rate of therapeutic antibody development, but also to make this entire development pipeline more efficient. And one of the proposed methodologies to be used in this setting to support drug development has been machine learning, because it could support this drug development pipeline in finding better molecules in a more efficient manner by reducing the reliance on experimental screening. So machine learning is a set of algorithms that learns patterns by approximating a function from samples to labels. To be able to use machine learning, one needs to have a sufficiently sized data set to be able to learn patterns and predict labels on unseen data. In protein or antibody engineering, this would typically be represented as having a protein amino acid sequence and using functional labels to predict. So to make the amino acid sequence, let's say, kind of machine readable, it needs to be encoded as numerical features, which could either be done as one-hot encoding, a binary representation, or other continuous representations of this amino acid sequence. Depending on the label that one wants to predict, one can use classification algorithms, for example, to predict binary labels like binding or not binding, or one can use regression models to predict continuous values, such as, for example, affinity or other developability properties. To be able to evaluate a machine learning model, or kind of like be able to tell how well this model actually works and how well it can predict on unseen labels, typically people use cross-validation, where you split your existent data set of samples or of sequences and labels into training and test sets. You train the models on the training set and you predict the labels of the test set. Eventually, you can compare the predicted values with the true labels and can calculate some kind of evaluation metric, which essentially tells you how accurate your model is able to predict the unseen or the labels for this data set. And this was essentially kind of like the motivation behind the project that I'll be talking about today, which focuses on the antibody engineering, so kind of like the second step in the therapeutic antibody development. And it was about precisely engineering antibody affinity with machine learning. So as we now know, machine learning models require sufficient amounts of labeled training data. In antibody affinity engineering, typically people use a high throughput method based on flow cytometry, which can generate large amounts of sequence function labels, but usually they're binary. Then you can train a binary classification model, and overall, very often or most of the times, the goal is to maximize the antibody's affinity towards the target antigen. However, in some cases, maximizing affinity is actually not always favorable. And this has actually been shown with agonistic immune modulatory antibodies that bind to TNF receptors that downstream or activate immune cells and that mediate or activate cancer cell killing. So this is being used in cancer immunotherapy. In this study, that I'm referencing here has actually examined the relationship between antibody affinity and agonistic potential of certain antibody variants. And they were actually able to show that lower affinity antibodies lead to prolonged survival rate in cancer mouse models. So therefore, we were we wanted to develop an approach to engineer antibodies across a range of affinities. So KD values that can be measured by binding curves from surface plasmon resonance or bilayer interferometry are much more accurate measurements of antibody affinity. However, and they yield to continuous data or continuous KD values. However, these methods are substantially reduced in their throughput because you cannot do pooled screens as opposed to flow cytometry. This continuous affinity data, however, would enable us to train machine learning regression models and to predict continuous affinity values and engineer an antibody variant across a range of affinity. So to generate such an affinity data set in this project, I worked with immune repertoire or antibody repertoire data that was generated from some colleagues or former colleagues in our lab. They immunized animals with a model antigen, hen lysozyme, short HEL, and they sequenced the bone marrow plasma cells, which yielded a data set of 83,000 VDJ sequences. In addition to this large antibody bulk repertoire data set, they also were able to identify an initial antigen-specific variant. So this was essentially a starting point for my project, because we thought that an antibody repertoire data set is actually like a really good proxy for selecting a smaller data set for affinity measurements, because the chances are probably higher in finding antigen-specific sequences within a repertoire data set that comes from immunized animals. 83,000 sequences, however, is like a way too large of a data set to be able to screen for affinity. So we aimed at selecting sequences from this data set, and we determined that 50 sequences, or roughly 50 sequences, would be feasible to screen in a single batch with the methods or the platforms that we had available at this time. So these 50 sequences we aimed to express and measure their affinities to be able to train and evaluate a number of fairly rather simple machine learning or regression models, such as linear regression, Gaussian processes, kernel-rich regression, and random forest. And the goal was essentially to use those machine learning models, or the trained machine learning models, to design new variants that would exhibit affinities across a certain range. So when we started with this project, we were thinking, or we were aware that the sequence selection process is quite a critical aspect, because obviously not every single sequence in this antibody repertoire data set will be specific to this particular target antigen that we were interested in. So therefore, we kind of split the sequence selection workflow in two, or with two objectives in mind. We split this selection workflow based on two regions of the antibody VDJ sequence. So first, we wanted to select sequences that are antigen specific. And to this, we first filtered similar sequences to the initial antigen specific variant based on 80% similarity. So we were looking to have a kind of like very stringent selection based on CDR3 region or CDRH3 regions. And in the second part, we essentially pulled out all of the VDJ sequences that contained those CDRH3 sequences. And we wanted to, and for this part, the goal was essentially assuming that those sequences that we've selected previously hopefully would be antigen specific. We wanted to select in the second part a broad representation of this sequence, of this antigen-specific sequence space. And therefore, we selected, or we applied K-mediates clustering and selected the cluster centers as variants that we went forward to express and characterize. So we used a mammalian expression platform and measured the affinity of those antibody sequences, which yielded a data set of 35 VDJ sequences with corresponding KD values towards the initial target antigen. We did some preliminary analysis as represented here as a phylogenetic tree of the amino acid sequence. And we wanted to see if there is some kind of relationship between or a clear relationship between the sequence and the affinity values, which we could see already in this plot, which also made us quite confident to go ahead and train these machine learning models and evaluate them. So in these so here you can see the cross-validation results represented as correlation plots of the true affinity values and the predicted affinity values. And as you can see, those correlate really well, which was really exciting to see, because despite this very small data set, all of the model or all of the nonlinear models that we trained could actually fairly or pretty accurately predict antigen affinity. We excluded linear regression from this evaluation because linear regression was not able to kind of capture the sequence function relationship. So looking a little bit closer into those model comparisons, we can see that overall all of these models perform fairly similar. However, we determined, or we see that the Gaussian process model trained with a matern kernel actually performed best. So now that we know that we can, at least within this data set, accurately predict antigen or antibody affinity, we wanted to see if we could actually use those machine learning models to guide the design of novel variants. And to test this, we designed an in silico library based on the existent mutations in our data set of the top two highest affinity variants and the lowest affinity variants and the initial antigen specific variants. We generated every possible single and double point mutant sequence that would not be in our training data set and not in the antibody repertoire data set. So for all of these synthetic variants now, we use the Gaussian process model to generate prediction or affinity predictions. And based on those predictions, we roughly randomly selected sequences across the entire range of affinity. When we then went ahead with experimentally validating those variants, we could actually see that for 7 out of those 8 sequences, the model was able to accurately predict antibody affinity, which was really exciting to see for us. So as a summary, to this project, we were able to select and characterize 35 antigen-specific variants from antibody repertoire data. The machine learning models, despite this very small data set, could accurately predict antibody affinity. And we were able to, for 7 out of those 8 in silico designed variants, or 7 out of those 8 variants exhibited the desired affinities based on the machine learning model predictions. So I have one last slide where I kind of wanted to go over a little bit of the aspects that I feel like I could not necessarily entirely address in this project. It also kind of relates a little bit to Eric's talk, which are probably also some kind of like open questions that I have, and maybe give some aspects to think about also for the discussion. And first of all, this data set is extremely small, and I'm also kind of curious because we were able to fairly accurately predict affinity. So that also kind of made me think that we probably didn't entirely push the boundaries of generalization of these models despite this small data set. So I'd be kind of curious to see how does predictive performance scale with the covered sequence space, as well as its corresponding affinities and a data set size. And also kind of relating a little bit to the aspects of like large language models, are there maybe different sequence representations that we could use in this particularly small data set setting that would improve generalizations? And I know people have used language model embeddings for this task, which with more or less success, but that's something to think about. And since we use Gaussian processes, these are probabilistic models. I'm also kind of curious whether we can actually use probabilistic uncertainty for antibody engineering, and how we can use this. Because when I did some kind of like small preliminary analysis, I was not necessarily able to find, let's say, an interesting enough correlation from those uncertainty estimates that come out of the Gaussian process predictions. So last but not least, I want to thank you all here again and for giving me the space. I'm happy to I'm really happy that I'm able to present my work here. I obviously want to thank Sai, who was my PhD supervisor. Thanks to Simon, Daniel, and Derek, who were very much involved also in doing this project. And of course, also thank you to the entire Ready Lab, who are a really nice environment and who also kind of helped a lot with my research.
Thank you, Lena, for this very nice talk, and thanks for staying on time. And Eric has a first question.
Hi, very interesting talk. Can you talk about the mutations that you introduced? Can you go back to the slide?
Yes, it's here.
Yes. So let's see. So the mutations that you introduced, um, you're saying that each individual mutation was never observed in your training data?
No, I'm sorry. So the individual sequences were never observed.
I
We
see.
use here only existent mutations from our training data set. So
Okay. And then you're just like figuring out what are the combinations of those mutations that will give high affinity. Okay.
exactly.
Great. I just want to double check that. Yeah, and it's interesting. Yeah, I would be interested in. I understand that linear regression didn't perform very well, but I mean, in some cases we've seen linear regression plus a single nonlinearity doing well. But I mean, I'd be interested in the type of sort of epistasis you see in these settings, but maybe I'll just have to read your paper. Thank you.
Yeah. I mean, I did actually, so we didn't really include linear regression because overall the metrics were really bad. But when you look at the correlation plots, you can actually see that some of those sequences line up really well and the other ones are extremely far out, which probably also kind of skews the predictions. But in the end, we decided to just exclude it because it obviously was not able to capture a decent number of the training data set sequences. Yeah.
Thank you.
Can I ask a question? So you mentioned how you chose those sequences that you screened experimentally, but you said they were from a bulk data set. So how did you add the light chains?
Yeah, so this actually comes, so this is like a model antigen.
Yeah.
And I actually did not myself choose the light chain, but we use the light chain. We use a constant light chain, so one light chain for all of those variants. But this light chain was known to be binding with this initial binding variant. So essentially, I took this initial binding variant that they already had found, and just took a constant light chain and only mutated or only put in some like the VDJ sequences that I selected.
And then just a general question. So why do you, because I think you seem yourself surprised that it worked quite well. Why do you think such a small data set was sufficient to predict affinity?
I think overall, all of those sequences are fairly close to each other. So
Yeah.
I have to like think back. I did some analysis on really just kind of like comparing the edit distance and like on the entire VDJ region, which I've probably came up with like up to 10 amino acids or up to 10 edit distance, which is still probably fairly close together. But yeah, I think it's probably like a matter of like, yeah, a matter of like a good selection, which was very like done in a very unsupervised manner, which I guess kind of like was lucky enough for me.
Right.
Yeah, but I think also like that the proxy of having this bulk repertoire data set from immunized animals is particularly useful. And while we did not exactly kind of select a clonal lineage, but selecting for 80% similar CDRs, like we are getting fairly close to that. But we do see that like more than 90% of the sequences have the same VDJ pairing, but it's 90%. So there's like 10% of sequences in there or a few sequences in there that do not fall into like the clonal lineage definition.
Mm. Mm. No, that's interesting. And regarding a bit also Eric's question about epistasis, so I'm not sure if you checked it, but if you have two mutations that separately would increase affinity, if you combined them, did they increase even more affinity or yeah?
That's a good question. I never really looked at it in like this kind of sense. I mean, I looked at the sequences itself. It was like in total like 50 sequences, but like I didn't do any statistical analysis of whether there is any like direct or actual epistasis. But that would be kind of interesting because in the end, like this would kind of explain it. But then also like I guess if it was only epistasis, maybe the linear model would have also performed better.
Mm.
Yeah, should have actually, right? But yeah.
And do you think that because there is kind of a something in the literature that so because before deep mutational scanning, people thought, oh, there are many so that the epistatic landscape of proteins is very complicated, but it seems like there are more and more papers coming out that it's actually not that complicated, and your paper kind of is one more point towards the non-complicated field. But do you think it's maybe also antigen dependent, that if you had chosen another antigen, that it would have been not so? So is it kind of epitope dependent? Because I guess all your antibodies bind similar epitope, which I think you haven't checked, but I think we could assume that. Yeah.
I mean, I agree. Like, I think it's a very good point. I also assume that they bind to the same epitope because they're also similar. But I don't know. I have no means to, or I had no means to check. We only like checked for affinities. But this might be, I think, a really good point, and also like relating to all of those like demutational scans, like using flow cytometry, where people generate these like very, very large data sets, which is like quite amazing. But at the same time, like there's no way for you to check like whether those all bind like very likely they bind different epitopes and they bind like the antigen from all different kinds of angles. So I think considering epitope is something that is actually quite important, and I think this also like very very much simplifies the task, which probably was the case for this project for me here. Yeah.
Yeah, I agree. Yeah. No, I think it's very nice. Are there more questions for Lena? No? Then thanks, Lena, very much. It was really nice. Thanks, Eric, again, and thanks everyone for coming to the talks. And they will be online in a few days. Thanks, everyone. Bye-bye. Bye-bye.
Thank you,
Thank you so
Victor.
much.
Bye
Bye.
now.